Welcome friends, welcome to Datta Biology class. Let us continue our discussion on transcription. This is the part 3 of transcription, wherein we will discuss from RNA how proteins are decoded. This is a complex one. Attend careful attention, please pay your attention, and it is very easy. But once you learn it, then it will be easier, otherwise it will be a difficult one. So first, the complex process, say this is mRNA. Clear? Now mRNA, we have seen there are codons. How? This is mRNA. Sorry, this is not mRNA. In mRNA, this T is replaced by U. Uracil. So this is mRNA. In mRNA, these codons, these are all codons. I will come how this is converted to protein. This conversion is known as translation. In translation, we have seen from English to Hindi translation, from English to Bengali translation, from English to Marathi translation. So there is no relationship with these codons and with that of the protein. So it is translated. How this is translated? This is a very big story. Last century or before last century, in 20th century, so many scientists have contributed, like Nirenberg, our Hargobind Khorana, they have contributed to decipher these codes. How this codes is getting converted into protein. That we will discuss today. They say, these scientists, they say, if you start reading the nucleotides, how many nucleotides are there in RNA? Not clear. Nucleotides are in this way, say for example, this is adenine. Adenine joins with sugar. Sugar joins with phosphate. So adenosine monophosphate. This is adenine. When joins with sugar, adenosine, and then when joins with phosphate, adenosine monophosphate, or this is a nucleotide, deoxyadenylate acid. In case of RNA, it is not deoxy, it is riboxyadenylate acid. So adenine, guanine is a base, nitrogenous bases, joins with sugar and then with phosphate, adenine, guanine, thymine joins with sugar, then phosphate, adenine, guanine, thymine, cytosine. Cytosine joins with sugar, which we have come across in DNA. Sugar, then phosphate. So this is a sugar phosphate backbone, and these are the nitrogenous bases. So there are 4 nitrogenous bases in DNA and 4 nitrogenous bases in RNA. So these are all nitrogenous bases, or we'll say nucleotides. These nucleotides are available. There are 4 nucleotides available in RNA. Out of these 4, scientists have discovered that if you start, it will start from 5 prime to 3 prime. If you start reading the code from this 5 prime to the 3rd prime, so it will be like this. This is the first codon, first nitrogenous base or first nucleotide. This will be the second nucleotide, and this will be the 3rd nucleotide. If you start reading the nucleotides from the 5 prime of RNA, you will find there are 3 nucleotides. Scientists, after doing so many experiments, they have deciphered or decoded that these 3 nucleotides or nitrogenous bases are responsible for conversion from this to amino acids. How? Now, let us suppose we have gone through this. This is tRNA. This is 3 prime, this is 5 prime. 3 prime has 1, 2, 3 nucleotides, and they are called CCA. CCA. This is unpaired because it starts from here. 5 prime starts from here. This is unpaired. And there are 4 places where the nitrogenous bases remains coiled or remains joined with one another, because the nitrogenous bases have the affinity to align itself or join itself with one another. So AUGC, not ATGC, AUGC remain attached, and these are the sites. These I have said that this is protein or amino acid binding site. This is amino acid. This is amino acid binding site, and this is anticodon site. I have told you on the other day. If you see here, there are 3 codons. Not getting okay. Here, there are 3 codons. These codons are complementary to this. I told you, A will be here. It is RNA. So T will not be there. It will be U. Clear? U will be, U means T, will be A. UAGC. So this is known as anticodon. This is the mRNA codon on mRNA. This is the codon on mRNA, and this is the anticodon site. This anticodon site is not will not decide the amino acid, but this codon will decide the amino acid. How? Now this 3 codon AUG means for methionine, amino acid known as methionine. How this methionine is coming? I told you that within the cell, in the cytoplasm, so many amino acids are present. There are 20 amino acids. How these amino acids are going to the cell, coming to the cell? You are taking protein, meat, egg, vegetable protein as well as animal protein. You are taking proteins are breaking down into the intestine and it is being absorbed. So proteins are broken into amino acids and are carried through the blood to each and every individual cell. So in the cell there are so many amino acids, at least minimum 20 amino acids are there. Okay, these 20 amino acids are being collected by this tRNA, which will come during our transcription, because transcription process we have not started. In the last step and the next day we will start the transcription. If you know this, then that will be easier for you to understand. Now tRNA, transfer RNA, these are available in the cytoplasm. tRNA will pick up this methionine. tRNA will pick up alanine. tRNA will pick up valine. Or 20 amino acids they will pick up from the cytoplasm and they will come to this mRNA. mRNA will attach itself with the ribosome, because the protein factory is ribosome. Ribosome has 2 units. I am coming in the next. This is a unit. This is a larger unit. This larger unit. This mRNA comes here. mRNA remains here. So this tRNA is picking up from the cytoplasm and unloading the amino acid here. So tRNA acts as an packers and movers. So it is packing amino acid from the cytoplasm and delivering the same to the ribosome, the protein factory. How protein is produced? We are in the process. Now this mRNA have this codon. Scientists have discovered that the first, second and third nucleotides are responsible for production of protein, for decoding this to protein. If it is this protein. If it is something else, that protein. If it is something else, that protein. I am coming. So what I have said, that if you start your counting from 5' of the mRNA, the first nucleotide, the second nucleotide and the third nucleotide will constitute codon. It is called triplet and it is actually called the codon. This triplet will decide that which amino acid will be here in the tRNA. So tRNA have the ability to identify that where I am joining, where I am attaching myself. So this tRNA is the packers of this, and they have come to mRNA here for production of protein. Now, you say here the mRNA sits here. This is the smaller subunit of mRNA. Here if you see the codons 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3 and another 1, 2, 3. So these codons are there in the mRNA. How it has come into the mRNA? Because mRNA is produced from DNA, on the direction of the DNA, this mRNA has been produced. If you find here AUG, if you find AUG, so this tRNA will have what? Methionine. This will be methionine. If you find UUU here, UUU, then this will be tRNA is there. This tRNA, this is tRNA. We have anticodon side also. These are anticodon side. There is no linkage between anticodon and production of this. Anticodon is binding side. Anticodon binding side. It is a binding side here only. So we consider this one. In mRNA, whatever is there, that will constitute the production of amino acids. Clear? Now, UUU will produce phenylalanine. This will produce phenylalanine. So, methionine is produced and then phenylalanine is produced. This methionine. Methionine is produced, then phenylalanine is produced. If you find GAU, leucine, if you find GAU, I am coming why this is GAU is methionine, that I am coming, that will be much more easier. Okay. Now this tRNA, tRNA is there, GAU will produce leucine, gRNA, AAA, lysine, GAU, sorry, CUA. That will be produced. UUU, then AAA will be easier. But AAA. Say here it is AAA will produce lysine. So here lysine amino acid will be produced, CUA. Then here if in mRNA it is CUA, then the tRNA will produce another protein, CUA is leucine. CUA is leucine. So, one by one, amino acids are there. Whenever it will come, whenever we discuss on ribosome, we will say, and during our process of transcription, we will find how the 30S and 50th subunits joins and how it is being produced. Now, let us confine to this, that if the codons are there, 3 triplet code scientists have discovered, that if it depends on the 3 codes, not the 4. So, A, G, U, A, G, C, A, G, C, U, like this, so many codes may be there and coming. There are 64 codes. Okay. Now, these amino acids will be produced. And these amino acids will join one with another by CO-NH bond. CO-NH bond. CO-NH bond. These bonds are known as peptide bonds. These bonds are known as PPTID, peptide bond. So with the help of peptide bond, the amino acids are being produced. This is amino acid, amino acid, amino acid, amino acid. These are joined with peptide bond, and these forms a protein. This protein, from this protein, why this protein are produced? These proteins are antibody, hormones, enzymes, they are all made up of protein. So proteins are made up of linear amino acids arranged in a definite sequence. Methionine, phenol, lysine, leucine, say it will produce something, and some other things is there, it will produce some other protein. So in this way, the proteins are being produced. Scientists have discovered this. Now, based on the variety of experiments, we have come across that, or scientists have come across that this triplet code can produce so many codons. There are 64 codons can be prepared from this triplet. 4 into 4 codon into 4. 64 codons can be prepared. I am coming that way. That is available in your books, but we do not go into the deep of the chart. Let's see. Please keep it in mind, this is the first position, this is the second position, this is the third position. There is no fourth position because they have discovered that triplet code is sufficient to produce amino acids or to produce protein. Now, to know it very clearly, you have to know 4 things. You have to keep it in mind 4 things. Because I will not tax you with remind this, keep it in mind nothing. You keep it in mind one thing, that is U, C, A, G. U, C, A, G. Here also U, C, A, G. Clear? You see it. Now, you multiply with these 2. U. Here it is U. Here there will be another U. There will be another U. There will be another U. And this C will have 4. 64 I am telling. This C, this is U. This is C, this is U. C, U. This is 1, 2, 3, 4. C, U. There are 4 nucleotides. U, C. Now A multiplied it with U, A, U. Then A, U. Then A, U. Then A, U. And for G, G, U, G, U, G, U, G, U. G, U, G, U, G, U, G, U. So in the mRNA, this is the first position and this is the second position. First position, second position. And in the last position, U, C, A, G. U, C, A, G. U, C, A, G. U, C, A, G. These are completed. Now, in the next one, what I will write? U and C. So all these will be U, C, U, C, U, C, U, C. Then this is C, C, C. It will be C, 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 C. First position, second position, cytidine, and C, C. This will be A, adenine with cytosine, A, C, A, C, A, C, A, C. And the last one will be G, C. So it will be G, C, G, C, G, C, G, C. Now you simply write U, C, A, G. U, C, A, G, you have to keep it in mind. U, C, A, G. U, C, A, G. U, C, A, G. Oh. Now U and A. So it will be U, A, U, A, U, A, U, A. Now C, C, A. C, A, C, A, C, A, C, A. Just multiply A with A. It will be A, 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 A. G, G, A, G, A, Write U C A G. U C A G. U C A G. U C A G. Clear? Now what I will write? U and G. If I multiply it, it will be U G. U G. U G. And U G. C G. It will be C G. C G. C G. C G. A G. A G. U C So, with the help of this triplet, and since there are 4 nucleotides, so 4 into 4, 16 into 4, 64 codons scientists have prepared. 1, 2, 3, 4, 1, 2, 3, 4. 4 4 jar 16 and 1 2 3 4 16 into 4 jar 64 codons scientists have discovered. Out of this, you have to keep in mind only a simple thing. Please write AUG. AUG means August. How you will keep it in mind? You simply keep it in mind that it is in the month of August they have discovered. This AUG is meant for the amino acid methionine. AUG for methionine. Clear? August, AUG for methionine, nothing more. AUG for methionine, and 2 or 3 more. It will be noticed that 1, 2, 3, 4, that is 3, 4, I have written 3, 4, and here 1, 2, 3. So 3, 4, 3 you have to keep in mind. What is that? 3, 4, 3. Here also 1, 2, 3. That is 3, 4 and 3. These 3 are previously called nonsense codon. Nonsense codon, or today these are called stop codon. STOP, stop codon. Because when mRNA is producing protein, the starting point will be August. So it starts with methionine, and there is codon here. Say for this UAA is there, this codon is there, the mRNA stops. Because it has no protein on its side. So these are called nonsense or a nonsense codon, or these are known as stop codon. If it is UAG, then also it will stop. If it is UGA, then also protein production will be stopped. That is, from this point to this point, this protein is meant for something, production of hormone, production of enzyme, production of whatever it is. So these three you have to keep in mind, that August is the starting, and 3, 4, 3. These three you can keep it in mind, or if you write like that, it will be easier for you. It will be 3, 4, 3. It is 1, 2, 3. It is 4. It is 1, 2, 3. 3, 4, 3. These three are stop codon. Now, why I am saying this? Because you see, this all means for amino acid written in your book, glycine. The glycine is produced. All these codons, if they are GGU, glycine will be produced. So it has 1, 2, 3, 4 codons. So 4 glycine will be produced. So it is the first codon and second codon it decides, and third codon is known as Ubit's codon, that we will not discuss. So this will produce glycine, alanine GCU. These 4 will produce alanine. It is written in our group, alanine. This is threonine, ACU. Threonine, THREONINE, threonine. So NIA, this proline serine. This all will produce proline, PROLINE, proline. This will produce serine, ACRINE, serine. So in this way it goes on. This will produce valine. Scientists have discovered that they are producing these amino acids. There are 20 amino acids. If you go through, I am writing it down. AUG is methionine. It is available in your book. These are called isoleucine. Isoleucine. This is leucine. 1, 2, 3, 4. LEUCINE, leucine. And it is, these two are leucine. LEUCINE, leucine. These two are phenylalanine. Phenylalanine. And GAU. These two are aspartic acid. ASP, aspartic acid. These two will produce glutamic acid. Glutamic acid. Look, these are all non-histone protein because they are acidic in nature. They are having more COOH. And glutamine, histidine, CAU. These two will produce histidine. Histidine, tyrosine, this will produce tyrosine. Tyrosine, it is available in your book. You can go through the books. This is tyrosine. This is also tyrosine. This is cysteine. These two will produce cysteine. Arginine. These will produce arginine. ARGININE. AGU and serine. These two will produce serine, and these two will produce arginine. ARGININE. You go through your books and you will find there are 20 amino acids is produced. 20 amino acids. If you go through all this, you count it, and there are twice. Here also arginine, here also some arginine here. So if you collect it and assemble it, you will find that 20 amino acids are being produced. In the 28th century, Hangal, Purana, Starnbal, and Nirenbal, they have discovered this. They got the Nobel Prize for this. So they have deciphered the mRNA triplet. They deciphered And by experiment, so many experiments, they have found out that these proteins are being produced in a linear manner, and it is in a sequential state. They have produced all these amino acids, and these amino acids are in a sequence. This is RNA to protein translation. RNA to protein translation. And we will go to the transcription process, the exact process on the next episode. Achha, prior to that, let me say one thing here. You will take it down, or you go through your books, you will find it. And simply you write in this way: first position, second position, and these are the UCAG is the third position. UCAG, write UCAG last, and in the first you just multiply like Mendel dihabet experiment, what I have seen. Now we will discuss one thing, only 5 minutes or 2 minutes will be taken. Why these proteins are produced in a sequence? If the sequence is disturbed, then what will happen? What will happen if the sequence is disturbed? All of you have heard in my RBC classes. Please go through, feel free to go through my RBC classes. There I have said sickle cell anemia. Sickle cell anemia. In sickle cell anemia, what happens? The RBC becomes sickle shaped. The RBC usually like this, biconvex, and it do not have any mitochondria, nucleus, anything to accommodate more and more oxygen. So more and more oxygen comes here in RBC, producing oxyhemoglobin. So you have heard the name hemoglobin. In hemoglobin, is constituted of two parts, that is heme portion and globin protein portion. It has 4 Fe. So like this I am writing. In hemoglobin, this is heme, and these are the globin portion. In hemoglobin, there are 4 chains, 2 alpha chains and 2 beta chains. Clear? In hemoglobin is a conjugated protein. It consists of heme and globin portion. There are 4 Fes. They carry the oxygen. Because they carry the oxygen, these are known as oxyhemoglobin or deoxycarboxyhemoglobin, whatever it is. So hemoglobin consists of 4 pyrrole. These are called pyrrole rings. It remains in this way. Some pyrrole rings in this way in 4 types. So I am concerned with 2 alpha chain and 2 beta chain. These 2 alpha chain contains 141 amino acids. 141 amino acids. Clear? So this chain contains 141 amino acids from mRNA. It is being produced 141 amino acids. It also contains 141 amino acids. And beta chain contains 146 amino acids. 146 amino acids. In these polypeptide chains, 146. These amino acids are there. 2 alpha identical alpha chains, 2 identical beta chains are there in hemoglobin. thyroid rings, then these chains are. So total it comes 500. It contains 674 amino acids. So hemoglobin is made up of 674 amino acids. And these amino acids are arranged in a sequence, in a definite sequence, in an orderly sequence in these 4 chains. Now, if you go through its beta chain, this is also beta chain. You will find in the beta chain, the number 1 is valine, V-A-L-I-N-E, valine. Number 2 is histidine. Number 3 is leucine. Number 4 is threonine. 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 Number 5 is proline. Number 6 is glutamic acid. So, I have written out of the beta chain 146, I have written 6 only. Clear? 140 I am keeping it aside. So, it has been observed that valine in beta chain, another one, valine is there, histidine is there, leucine is there, this there. So 1, 2, 3, 4, 5 contains 1 valine, histidine, leucine, threonine, proline. These are all same, but I am marking it as red. Here in 6th position, the amino acid is valine. So is it in a definite sequence? No, it is not in a definite sequence. Instead of glutamic acid, valine has been produced. You go through my earlier chart, you see glutamic acid is some GGU or GGA. So there is the defect production of this. So this is a sickle cell. Because of this presence of valine in the 6th position of beta chain of hemoglobin, the RBC becomes sickle shaped. This, because of this sickle shape, it cannot carry oxygen, and the negroes, this is prevalent in negroes, they suffer from anemia. That anemia is known as sickle cell anemia, because in the beta chain of what hemoglobin, in the 6th position is one of the beta chain is substituted by valine in case of glutamic acid because of this production of amino acids. So amino acids are produced from mRNA. These amino acids are produced. 1, 2, 3, 4, 5. These 5 have been produced, and the 6th position is produced valine. So it is not in a definite sequence, and because of this indefinite sequential arrangement of amino acids, the RBC becomes sickle shaped in negroes, and it is a genetic disorder because of this. Clear? So that constitute the end of today's episode, and the next day we will start a little bit of ribosome. Ribosome, it will take 2 or 3 minutes. So after completion of ribosome 30S and 70Y, 70S and what are there inside, we will discuss on the next episode, and on the next episode we will complete the transcription, the main process. Till then, take rest. Thank you.